Let's look at antibodies. We've been talking about them a lot. What are they? As I mentioned before, they are immunoglobulins, so they are a protein. Specifically, they are your gamma globulins in your plasma. You will need to know the basic anatomy of an antibody. They will have 4 polypeptide chains. Recall that a polypeptide chain is made up of amino acids, so we have 4 here. 2 of them are, you can see they're larger, so those are my 2 heavy chains, and I will have 2 light chains. Antibodies will have either a Y shape or a T shape. Each one of these polypeptide chains will have 2 regions. The variable region, you will see that here. The variable region will be distinct and unique. It is here at the variable region where the antigens bind. And remember, B cells are specific as to which antigen they will bind. So this is Y, and that's called the variable. So that's going to be unique and different for each type of B cell, and it will be for the antigen. And then you're going to have a constant region. And these constant regions are shared by all antibodies that belong to one class. There are 5 different classes of antibodies. Antibodies have multiple functions. The first one is precipitation. In precipitation, a soluble antigen binds with the antibody. These are cross-linked, and this causes the soluble antigen to become insoluble, making it easier for phagocytic cells to phagocytize the antigen. So it just precipitates it out of the plasma. Agglutination is similar in that you're going to have an antigen. In this case, if we're talking about blood, we're talking about the antigens that determine your A, B, O blood type, so your A and B antigens. They will bind with antibodies, and they will be clumped. So you're going to have one antibody that will bind with multiple antigens, whereas with the precipitation, you're going to have one antigen binding with one antibody, but they're going to be cross-linked. So with agglutination, you're clumping the antigens together, and this will increase or enhance phagocytosis, make it easier for phagocytic cells to eat the antigens. Lysis. Recall that lysis is when a cell bursts. This is related to complement. During the classical pathway of complement, antibodies bound to the pathogen, such as the bacterium, will activate the first complement in the classical pathway. There are 3 outcomes to complement, regardless of what pathway is used. Recall that one of them is the formation of MACs, membrane attack complexes, and these put pores or holes into the plasma membrane of the pathogen, the antigen. Water will move in by osmosis, and the cell, the antigen or pathogen, will burst. So this lysis is related to complement. In addition, you're going to have enhancement of inflammation. This can be done through complement, but also some antibodies will bind to mast cells and basophils and trigger them directly to release histamine. Opsonization, this is also an outcome of complement, but in addition, antibodies can function as opsonins. They can bind to the cell that is going to be eaten, the antigen or pathogen, and this will make it possible for phagocytes then to bind to the antibody which is attached to the pathogen so that they can eat it. Neutralization. Neutralization involves antibodies neutralizing substances such as toxins and venoms by covering the binding sites on these chemicals so that they cannot bind to your cells. And as a review, remember, antibodies and BCRs are exactly the same molecule.